Okay, I'm getting it now. The song was talking about kombucha. I get it.
My name is Jackie, and I am your not so quick to pick up on things MC for tonight's Science on Tap. I'm really excited that you guys came out on such a gorgeous evening to spend some time learning more about the amazing microbes that make all of those delicious food and beverage even more fantastic. Tonight's speaker is a phenomenal research scientist who comes all the way from Australia and has bring with him just some great insight into the story behind this amazing drink. So I am delighted to introduce Dr. Chris Curtin, an associate professor of fermentation microbiology. Yes, did you know you can major in fermentation microbiology? I am questioning all of my life choices. He's a professor of fermentation biology at Oregon State University, where his research focuses on looking at how the microbiome of cultures used for food and drink preparation have evolved over time. Now, if this is not the coolest thing ever, I don't know what is. Dr. Curtin comes all the way from Australia and has been in Corvallis for the last 7 years. And here is a crazy fun fact about him. He did not have any fermented foods growing up. So his love and passion for this area of food and drink science is a relatively new thing, and I'm so glad that he chose this as something that he wants to research and share with all of us tonight. So let's welcome to the stage Dr. Chris Curtin.
Thank you. All right. Well, it's dark, but what was the show of hands again for the number of people who brew their own kombucha? Nice, okay. Because, tonight I will share with you what I know about kombucha, but honestly, that's from a microbiologist perspective, and at the end of the day, some of you probably know a lot more. Maybe you can come and teach me some things afterwards, but I will share what I know about kombucha from a microbiologist perspective. And just to make sure we're on the same page, which I think most of us are then, we're talking about fermented tea, not tea made from seaweed, which is a very different thing and evokes a very different response, right? So, from a microbiologist perspective, kombucha is super cool because it needs yeast and bacteria. And I like to work on yeast a lot. I like to work on bacteria. Now I get to work on them working together as a community. And it's already been given away. Maybe it didn't get my accent, but it is definitely Australian, not New Zealand. Now, if anyone's from New Zealand, it's okay, you're cool too, but Australia's cooler. So as a bonus tonight, you'll hopefully walk away with a little better understanding of some Australian slang terms, which, if you happen to get the chance to run into Margot Robbie, then you can impress her. Now, I will say I'm not, I'll give you the first slang term. Genuinely very Australian is called ochre. I'm not super ochre, but, I use a bit of slang, so. Anyway, so I'm from Adelaide in Australia, which is in the southern middle part there. And we do have, we have a whole island called Kangaroo Island. And we do have kangaroos jumping down the street, but not as frequently as we find deer. And, oh, thank you very much. I was going to say we have it, well, sticking first with the kangaroo theme, we have a saying, a few kangaroos loose in the top paddock. If I do a terrible job tonight, feel free to tell me that maybe I've got a few kangaroos loose. If I get things wrong, that you know better. If on the other hand, and if I do a bad job, then it's my shout. But thank you, I don't have to shout myself a beer, I've got one right there. Now, if I do a good job, you might congratulate me with a "on your mate. You did a really good job, I enjoyed this presentation." And I should say though, if you go to Australia, that like many phrases, Australians find a way to use them sarcastically as well. So you could also, if you think I did a bad job, you could say, "On your mate, you did really good, well done." So I'll be looking out for that later. My sarcasm, senses are pretty strong. All right, sticking with the few kangaroos loose in the top paddock. When I first moved to Oregon 7 years ago, and yes, it's true, I grew up being exposed to very few fermented foods. I ate yogurt, that was about it as a kid, and honestly was probably not very full of live cultures. We're talking about the 1980s here. And when I moved here, I thought I'd landed somewhere a little bit strange. I'm not going to say that I thought everybody was loopy, but I walked into a supermarket and I saw a lot of kombucha, and I was like, wow. I'd heard of kombucha. I was already a fermentation microbiologist, so I was aware of it from its fermentation point of view, but I'd never actually, encountered kombucha in the wild in Australia. So I was very excited, and then I tasted it.
It really smells like a public restroom.
Probably everyone's first encounter with kombucha.
It smells like a public restroom. You know what? No. Well.
All right. Very much okay, here's another little Australianism. We'll say yeah, nah a lot, which just means yes, or I'm not quite sure. Sometimes it means no, but, yeah, nah. So, you drink some kombucha for the first time, you're like, wow, that's a lot going on. Takes a little to decipher. So, how did I actually get here then to talk to you about kombucha? Because I grew up definitely not drinking it. I didn't come here specifically to, start doing research on kombucha. I came here to do research on beer. I like beer. I like sour beer in particular. So how did this happen? And it all came down to I had a new student start, Keisha, who walked in one day and said, I want to do my thesis research on this, and brought in a SCOBY, and she says, this comes from kombucha, and it's what does the fermentation. I'm like, okay, I'm intrigued and a little grossed out. Thanks for bringing that in, show and tell. And then, here's the Stranger Things reference, but, if you just brew some tea and have some sugar in it and then leave it out, it will just happen, right? You will form a nice little layer at the top, and that's a baby SCOBY forming, right? So I thought that was pretty cool too, very strange. And then you see pictures like this, and you're like, wow. I'm not sure I want to drink that, but okay. Again, a lot going on in there. Now, those of you how many of you got this question correct in the trivia? I hope at least everybody who makes SCOBY at home. SCOBY stands for Symbiotic Culture of Bacteria and Yeast. It's definitely that weird-looking, slimy stuff. And at this point, I was in my student had come in, show me the SCOBY, talk to me about kombucha. It's like, there's a lot of different yeast and bacteria. The research community is confused as to which yeast and bacteria are there. So there was a lot to work out. So I was in. Another Australianism. I felt the need to, therefore, to suss it out. I had to work out what was going on inside this strange fermentation. Coming back to, questioning choices in life, I enjoy being a fermentation microbiologist because I don't have to work on poop, which a lot of microbiologists, that's whether you're working on the human microbiome or you're working on chickens or cows, you often end up as a microbiologist working on poop. So, cool, I don't have to do that. And in terms of, thinking about choices in life, I should have just, when I came here and saw all that kombucha, gone straight back to Australia, opened a kombucha company, and I'd probably be a multi-millionaire by now, but I'm an academic and we don't make the best choices all the time. So anyway, and the reality is, you look at this projected growth and I should probably just do that anyway, right? There's still plenty of opportunity out there, and those of you who are making kombucha at home, you certainly could go ahead and start to scale that up. Later on in the talk, and I'll get to the not so happily ever after part a little bit later, is that there are some teething problems with trying to scale up and make kombucha that is good and that complies with that alcohol limit. So we'll get to that bit later. Anyway, why else am I working on kombucha? Because it's crazy popular and continues to grow in popularity. And a big part of that, what's driving people getting very excited about kombucha, at least part of it anyway, it's not that it tastes bad, right? I mean, there are a lot of really tasty kombuchas out there, and then there are some that are pretty funky, and really just depends on your first experience whether you're going to buy into the flavor or not. But kombucha is part of the broader trend surrounding gut health and fermented foods. So, number one for 6 years in a row, fermented foods as voted as the top superfood amongst dietitians. All right, and what's driving that? The fact that we consume kombucha and many of these other fermented foods containing live microbes. So, and more specifically, the fact that those live microbes are capable in some way of exerting a positive influence on our overall health. And I will talk about that a little later. You know, we've certainly come a long way from the days of just germs are bad, right? Which is certainly what I was taught growing up, you know. Well, I mean, convincing kids to wash their hands takes a little bit of work, and you know, we should all wash our hands because that's what happens if you go and touch something and then put your hand on a petri dish. So who shook hands on their way in today? You know, you've just shared your microbiome, and that's okay. There's nothing wrong with sharing, because at the end of the day, the microbes that are on us, that surround us, many of them end up inside of us in our gut. And there are some that, you know, we don't want inside our gut. Enteric pathogens, right? We don't want those. But a lot of other microbes perform very important functions inside our gut and help us to function and be healthy. For example, helping us to fully digest our foods, help our immune system to kill off those pathogens that do make their way into our gut from time to time. I've got a lot of my colleagues are food safety specialists and work on microbiology from a it's going to kill you point of view. But it's kind of funny because some of them are super lax when it comes to worrying about, you know, the cleanliness of foods, or, you know, do you need to actually pasteurize milk, things like that, crazy stuff. But or at least I think so. But you know, they're like, you know, honestly, our guts are super resilient as long as you keep them healthy. Like, you'd be surprised at we're ingesting salmonella all the time. We're just not ingesting enough of it to cause a problem. Anyway, I digress. The idea that, you know, food is an important part of health has been around for a long time. We're talking nearly 2,500 years since Hippocrates, you know, made some of these observations about the potential role of food in health. It's just today we're starting to get a better understanding that a key part of that is the microbes inside the food, and we're starting to recognize more of the effects that they can have. And one that's, you know, I remember the first time I started seeing papers about this and I'd be like, really? I don't quite get that. I get that microbes can be inside our gut and can change the rest of the microflora in your gut and maybe contribute to, say, reduced inflammation. And then I can see that that can be helpful for somebody like my daughter with ulcerative colitis, who I definitely recommend to eat fermented foods. The idea that somehow the microbes in our gut, through the chemicals that they make, are communicating with our brain and changing our overall mental health and overall health and the response of our brain to things that are going on in our gut, that's pretty crazy. But fair dinkum, this is for real. Some very good science and some very good scientists at OSU are pursuing some of these questions about the gut-brain axis and the role of microbes in our gut. All pretty cool stuff. So this is all to say, give your microbiome a bit of a hug because it does good things for you. Take care of it. You know, the reality is our ancestors ate a lot of fermented foods, and we, you know, in industrialized Western society moved away from consuming those kinds of foods, and probably for the worse. So there's a lot we can do to enhance our gut health, and fermented foods are definitely part of that. So, and they taste good. All right, so to recap, why am I here talking about kombucha, given that, you know, I'd never really seen kombucha until 7 years ago? Weird, number one, cool, I want to work on that, and it's not poop. So, you know, we're in my sweet spot here. It's not poop and it's interesting. And superfood, so it seems like something worth doing research on and trying to understand how it all works. Which brings me to the next part, and I am going to have a sip of this beer. All right. So this is the part from the microbiologist's point of view to talk about how the kombucha fermentation works and the not so happily ever after part of what can go wrong. So let's take a look at what's going on inside of a typical jar full of kombucha and SCOBY that's fermenting, what's actually going on inside there. You know, we have yeast and bacteria present, and the interesting thing is any given SCOBY, it could contain, even if we only looked at those that are there in, let's say, the top 10%, or top 90% of the population of the yeast and bacteria. There could be 6 yeast, there could be 6 bacteria there, all working together. We do surprisingly find that most of the time, it really boils down to a couple of key yeast and bacteria that we find every time. Further interesting, well, I think so anyway, the yeast in question, some of you may know, may not know, it goes by the name of Brettanomyces, or Brett for short. And Brett's what we find in sour beer. So there's some overlap there in terms of the impact on flavour and funkiness. It's also the same yeast that we find that spoils wine. So we don't want it in wine, but we do want it in sour beer and kombucha. All right, so the yeast and bacteria are working together to metabolize mostly the sugar that we put in. Because when you steep tea, there's not a lot there, right? Tea, at one point, before I realized I was going to talk for way too long, I had the Ted Lasso meme of tea being brown water, and why would you drink this? Right? I'm not a tea drinker, but it has phenolics in there. It has some nutrients, very little, but there's not much there to ferment. So we have to add some sugar, and then the yeast and bacteria can go to work. Once the primary fermentation is done, I'll go back through this in a little bit more detail in a second, but once the primary fermentation is done, and we remove the liquid from the SCOBY, and we want to put that in a bottle, and we might flavor it, usually not with, well, maybe you do, but not usually with really chunky stuff like that, but maybe a puree. We're still adding live microbes. So there are microbes that are moving from, well, they're still in the liquid, right? So they're now in that bottle that we've sealed, and we've dramatically changed the environment from an open jar that's covered with maybe some cheesecloth, now into a sealed container where we're trying to allow the microbes to perform carbonation. And it's a selective environment that changes which microbes can survive. So not all the microbes that were present in the SCOBY and in that primary fermentation are going to be there in the secondary fermentation. So as I said, primarily this one's just about making it a little bit fizzy. Now, as I'll get to later, one of the steps where things can go a little bit wrong if you leave it for too long. Now, I do remember something else fermented that I did have as a kid. My mum used to like to make ginger beer until she accidentally left a batch at the back of the pantry for a couple of weeks, and then discovered that my brothers and I had gotten into it. And we were, I think I was probably 9 at the time. My youngest brother was 5. Well, we were just a little bit drunk, but so that was the last time that we had ginger beer in the house that was homemade. A bit sad. All right, so we have this kombucha in the bottle, and now we're going to drink it and hopefully be happy, enjoy it. And some of the microbes are still going to be there, because the whole point of kombucha, well, one of the driving features of kombucha that makes it popular is the fact we're consuming live microbes. Now, how many of them, this one should really come with a question mark, how many of them make it to the gut and thrive there is a little bit open to debate, and is one of the more controversial topics that I'll get to towards the end of this presentation. Okay, so now I'm just going to walk back again to the primary fermentation and the secondary fermentation and talk about what's going on in a little more detail. Not too much detail. And this is where we see some microbial cooperation, because we have the yeast and the bacteria there. And in order to carry out this metabolism, where we're taking the sucrose, we're making ethanol, and some of that ethanol is getting used to make the acetic acid, right? We need the yeast and bacteria to do that. Yeast can't do it by itself. Bacteria can't do it all by itself and give us something that resembles kombucha. Right? Because otherwise, we're either making something that's alcoholic, just like a tea wine, or we're making a vinegar. And kombucha kind of sits in the middle. All right, so what are the yeast doing? The primary thing that they're doing to kick everything off is they are splitting that sucrose, because we usually just add sucrose in when we're sweetening kombucha, or the tea to ferment to kombucha. If you're making a jun and you're using honey, then it's a little bit different, because we have some other sugars there as well. The yeast is also fermenting the sugars to ethanol and carbon dioxide. Now, at this point, because it's all open at the top, the carbon dioxide isn't really that important. We're not trying to capture that carbonation yet. All right, this is happening throughout the fermentation. The acetic acid bacteria don't really like the sucrose. They want the yeast to, first of all, break that down and make the ethanol. Then the acetic acid bacteria are going to start to oxidize the ethanol into acetic acid. and they're going to oxidize the glucose into another organic acid called gluconic acid. And then we also have the lactic acid bacteria, which they're there, and they can perform a bigger role in some kombuchas. Really depends on how you're making it and how aerobic or anaerobic the environment is. Oh, now I remember what I was going to say about the acetic acid bacteria. They need oxygen, like us, right? They have to breathe. The yeast and the lactic acid bacteria don't care. They're happy either way. But this fact that they have to oxidize the ethanol or oxidize glucose to make energy is really pivotal and what shapes this microbial cooperation. Now, the other thing the acetic acid bacteria are doing, which is really important, as I'll get to why it's important, but they're making the floaty part of the SCOBY. They're actually constructing the raft, if you like, and they're doing that by making cellulose. So they're using some of the carbon that's derived from that sucrose and ethanol, and they're actually using that to make cellulose. Now, when you're making kombucha, the goal isn't to make cellulose, right? We don't drink that part. So you don't necessarily want the bacteria to be going to town and making a lot of it, but it performs an essential role, as I'll get to in a little bit. I'm just going to briefly show you a couple of clips out of this very long, very old video. It's just showing under a microscope what the acetic acid bacteria are doing. So inside the bacteria, there's this little spindle making fibers of cellulose, and it extrudes it out of its cell membrane, and the bacteria will just move backwards and forwards, essentially like casting filaments. So this, this starts to happen after a couple of days in your kombucha fermentation. I like this one because it's making a nice spiral. That makes a very ordered structure. It's one of the reasons why leftover cellulose from kombucha is a really interesting material for people who work on materials and alternative uses of materials, because it's a very ordered structure and can actually be turned into a lot of different things. One thing I will point out, that was an interesting one that flew past, if you do try and use some of your leftover SCOBY to make clothing, cellulose likes to absorb water, and we live in Oregon, so just keep that in mind. It has its limitations. All right. So as they're sitting there spinning that little web of cellulose, the bacteria are becoming embedded in it themselves, and they're trapping yeast inside that structure as well. And that structure floats to the surface, and remembering the fact that the bacteria that we're talking about here require oxygen, the purpose behind this starts to make sense. I'll just zoom in a little closer here and we'll look at what's going on. So if you imagine the bacteria and yeast, they're in the SCOBY, they're also in the liquid as well. In both places, the yeast are metabolizing the ethanol and the glucose, they're metabolizing the sucrose and glucose and making ethanol. There are also other nutrients present in this steep tea that are essential for the yeast and bacteria to grow, like vitamins, some nitrogen, not a lot. And this, because the SCOBY, this cellulose is a physical layer, and all of these other things are in the liquid, then there's a gradient from the bottom to the top in terms of the concentration. So there's more of the nutrients, the things that the bacteria and yeast need for food in the liquid than there is at the top of the SCOBY. And the inverse is true for oxygen. So the SCOBY is exposed to air, so there's plenty of oxygen at the top, and that oxygen is in a gradient as well. And so it turns out there's a sweet spot that's just below the surface of the SCOBY, where most of the action happens in terms of the acetic acid bacteria, in terms of them making acetic acid, and in terms of them making new cellulose. So those of you who have grown your own SCOBYs, I won't go all the way back, but you'll start off with a layer, and then the bottom part will start to fall away, right? Who's seen that happen? Yeah? So what's happening is the new layer is forming kind of towards the top, and it's floating more towards the top, and then the other part starts to sink as it becomes less active. So when you're, I'll get to that in a second, but when you're then reusing a SCOBY and it gets too thick, then the part that you want to keep usually is the newer growth, as I'll get to. Which makes sense when I'm about to say what can go wrong. Right, so one of the things that can go wrong is if your SCOBY gets too thick for the size of the vessel that you're doing your fermentations in, we end up with less of the oxygen getting, the SCOBY is getting too thick, so the bacteria are not getting enough oxygen, and they're not getting enough of the nutrients from the liquid. And what we see happen then is we'll get a stuck fermentation where we have too much accumulation of ethanol and not enough acetic acid. Now, when the SCOBY gets too thick, another problem that can happen is the top can dry out a little, which exacerbates this problem because cellulose, when it's dry, then it's actually a decent barrier for oxygen. When it's wet, the oxygen can go straight through. So if it's getting too thick, you get a lack of oxygen getting through. You can also get mold growing on the surface. And if that happens, you want to get rid of your SCOBY. No recovering from that. All right. So that's the primary fermentation, some of the key things that are going on. When we then, or one of the cool things, one of the reasons why kombucha is a very forgiving thing to make at home is that you don't have to start from scratch every time. You've got material from your last batch that you can use to start the next batch. We just make some more sweetened tea, throw in some of the liquid and part of the SCOBY. You might want to get rid of some of it, but you keep some of that as well. And that kicks off a new primary fermentation. And it will happily do that, right? And if you do everything consistently, you can get very consistent outcomes. If, I make sourdough, and I'm kind of prone to not being super consistent about which days that I bake, and therefore my feeding schedule is a bit all over the place. And, I know that's going to happen. Every now and then you just get a batch that is a little bit sluggish. And that's going to be true of kombucha as well. If you, leave it longer one time and don't ferment for as long the next time, or, you don't have your air conditioning set at a constant temperature, right? You're going to have some variation in how that performs. But generally speaking, it's a very forgiving system. And you can just repeat this forever. As long as you can keep that SCOBY healthy and not moldy, then you can keep making kombucha by reusing part of the previous batch. The rule of thumb is to reuse about 10% of the liquid from the previous batch. That does two things. It puts plenty of active yeast and bacteria into the sweetened tea, and it also helps to start off the acidification process because it's already acidic and you're introducing that into the tea, which is not. And that helps us get into a pH range where we don't have to worry about any weird things growing and causing spoilage. In the secondary fermentation, the key things that are happening there are the continuation of the yeast breaking down the sugar and fermenting it to ethanol and carbon dioxide. We don't really want too much ethanol, so that the idea here is to keep it to be a fairly short ferment. It's during this stage where you can also get more activity from lactic acid bacteria because they're not now having to compete with the acetic acid bacteria. There's no oxygen there, so the acetic acid bacteria are kind of done. What can go wrong here? Same problem, right? Too much ethanol. And this is the classic problem that has faced the kombucha industry. This is definitely the part that isn't always happily ever after, unless you want a boozy kombucha. If you don't, then this is what happens, right? There are numerous instances of kombucha companies being sued or having product recalls due to their non-alcoholic kombucha actually exceeding the level of alcohol that's allowed, which, as you know now from the quiz, is 0.5% ABV. Problematic if you're not meaning to be on the piss or hit the terps, or you're giving kombucha to your kids, right? So that half a cup, suggested amount starts to make sense. You can definitely drink too much kombucha just from the acidity point of view, and that's not recommended. But the other problem is definitely real here, that you have to be careful not to be unaware of the fact that it could contain alcohol. If you're a two-pot screamer, be particularly mindful of this and consume your kombucha carefully, and maybe not when you're going to be driving. Also, don't leave kombucha out on the bench at your house for like a day. that you've brought home from the supermarket and then remember to put it in the fridge, right? Unless, again, you're wanting a more carbonated, slightly alcoholic kombucha. All right, now onto the third part of what I'm going to talk about here, which is, is kombucha full of live microbes? Are they probiotic? This is where it gets a little controversial. And will drinking kombucha make me healthy and happy? And I'm going to talk briefly about some research that we did in my lab, where we specifically wanted to work out, well, what was in the kombucha that was available around us in Corvallis in Oregon to buy in January of 2021. So we went to the store and we picked out bottles of kombucha that fit 3 fairly common flavor profiles, the berry fruity kind, the more traditional herbal kind of flavors, or ginger citrus. And we ended up with 39 different products from 21 different brands. And what we were looking at was we collected both hard and soft kombucha because we wanted to compare what kind of microbes they had in them. So we had about 2/3 of the samples were soft kombucha and 1/3 were hard kombucha. And about 80% of the whole sample set made some sort of on-package claim about how alive or how probiotic the beverage was. And by that I mean these kind of phrases that you would see if you buy a bottle of kombucha. There's some really good ones here, like I really like the last one, bubbly probiotic tea for a happy gut. I mean, they're leaning definitely heavily into the perception there of what might be going on. Sometimes it can be a lot simpler. It can just say that it's raw, it's unpasteurized, live. So we have a whole range of different claims. Some of the samples that we came across actually name probiotics or name bacteria on their label. So here's a couple of examples here where there's a mention of a probiotic Bacillus subtilis, and this one's a little bit small and hard to see, but lists a few different kinds, and also mentions numbers like 1 billion of this or 4 billion of that. And who here takes a probiotic supplement? Anybody? All right. You'd be familiar with, there's always the mention of numbers like 1 billion, 30 billion. It's an arms race in terms of how many billions. It's worth noting that inside your gut you have about 100 trillion bacterial cells. So, a billion's like 0.00, I did this before, but I've forgotten how many zeros. It's at least 0.0001%, right, of the total number. Not saying that it's like a homeopathic amount that you're adding in 1 billion, right? And the reality is if they're the right probiotics, they could get inside your gut and grow. So we're not going to get too hung up on the numbers, although I'm going to talk about it. So who am I kidding? I'm going to get hung up on the numbers. But anyway, I'm a scientist. It's what I do. Anyway. All right. Hard kombuchas were hard. Like we measured the amount of alcohol in them. They had the amount of alcohol that they said they had. Okay, that's good. The soft kombuchas, on the other hand, about 1/3 of them, 1/3 of that sample set, exceeded the ABV level, and one of them had 4%. Now, these were samples that we took from directly out of the supermarket refrigerator, took them immediately to the lab in a cooler with ice, put them in the fridge, and then opened them within 2 weeks. So, take from that what you will. This is from 2021. I will say there's been a lot of movement in the kombucha industry to tighten this kind of thing up. Our study wasn't the only one that found this kind of problem. So it's a problem. And just to reiterate, if you're a 2-pot screamer, this is why, just be aware that the kombucha you're drinking could have more alcohol than you think. All right, back to the people who take some probiotics. Might be familiar with the term CFU. Like if you buy a probiotic supplement, it's going to say 1 billion of this, and it will talk about CFUs or colony-forming units. And that's what it actually means. It just means a cell or a couple of cells that might be joined together that if we put them onto a petri dish that they should be able to grow on, actually do grow. So it's a sign of life. So in microbiology, we're looking to count how many of these things we can find as an indication of how much life there is in that particular sample. 82% of the kombucha samples that we did this to had at least some CFUs, right? Some are not very many, but 82% of them would definitely meet the criteria of live. So if you're looking to drink kombucha and consume a live beverage that contains live microorganisms, 82% of the time that would be true. However, there are a few things that didn't quite stack up. The samples that said that they were live and or probiotic didn't necessarily have higher CFU counts. In fact, some of the products that made no claim had the highest CFU counts. One thing we did pay attention to was when the product listed the number, we compared the number that we got with the number that they had on the package, and it was always 10 to 100-fold lower. Keeping in mind that they usually refer to at bottling or at packaging, because that's the last time they can measure it, right? They can measure it, or they can dose in the amount that they want. Once it's out there in logistics and in the supermarket and in your house, they no longer have control over that. And the bacteria observed were not necessarily known probiotics. And this is, as I'll get to, not really that big a deal and not really that big a surprise. Fermented foods don't necessarily contain the small number of microbes that meet the criteria of being fully labeled as probiotic. Because to be considered a probiotic, it has to have been tested in a clinical setting and have proven health benefits at a particular dosage. And when we think about something like kombucha that could contain one of quite a large number of bacterial strains, there's simply no way that anybody has done all of that research. Now, with the exception, what we did find in terms of known probiotics were bacillus species, and these are the labels that I showed you before. Now, I'll draw your attention to this one on the left, where they say that they made this by, they took a traditionally fermented in-house kombucha and pasteurized it for shelf stability, and then they're talking about it having a probiotic in it. So in that example, it's very clearly obvious that that probiotic wasn't in the kombucha before it was pasteurized, it was added afterwards, right? Now, again, not necessarily a big deal unless you're hung up on the idea that you're drinking a beverage that contains the microbes that were in the fermentation. And even this other example on the right where they talk about specific numbers and they call out like they have Bacillus coagulans GBI 306086. They for sure don't have a way of analyzing, or they don't routinely analyze their kombucha at the end of fermentation to check if it has Bacillus GBI, all of those numbers. So they had to have added that. And that's actually a fairly common practice and one that's growing in the kombucha industry, is to supplement kombucha after it's been fermented and before it's bottled by adding in a probiotic supplement. Again, not a big deal, but it comes back to what you're expecting, I guess. And I should say that Bacillus is not something that we find in Kombucha SCOBY. It doesn't grow or contribute to the kombucha fermentation, but it's a very good probiotic supplement and does indeed survive in the bottle and probably can make its way to your gut. But at this point, you might be ready to spit the dummy because I've told you that kombucha companies sometimes don't do a great job with controlling the alcohol levels, and maybe the probiotics that are in there didn't come from the culture itself. So what I'm going to finish with, though, is saying that let's not get too hung up on that and talk about fermented foods and kombucha within this category and what it can bring and what it can do. Let's not spit the dummy. Right. As I kind of mentioned before, it's a very high bar to jump to call a food probiotic fermented, because you have to be able to demonstrate that the specific strain that has probiotic properties is in there, which usually means you had to have added it and you have to have a way of making sure that it's the dominant microbe in that culture. So in fact, very few, and we have hundreds and hundreds of traditional fermented foods around the world, and very few would actually meet this requirement of a probiotic fermented food. If you want to know more, I definitely recommend looking at this resource. If you just look up that acronym, you'll be able to find it. They have a lot of good information about what probiotic actually means. The point I'm making, though, is does it matter? There's a reason why kombucha and other fermented foods are being voted year after year as a superfood and being related to positive health outcomes. And there's been some, surprisingly, in many cases, the clinical research is kind of lacking, but in this particular case, this is a study where some researchers at Stanford put together a very compelling piece of data to show what happens when you consume fermented foods. And they were actually comparing high fiber diet and a fermented food diet. And for the high fermented food diet, they actually found there was an increase in the diversity in the patient's guts. Interestingly, not necessarily from the microbes that were present in the food, as in they didn't necessarily find the probiotic that was in the fermented food in the person's gut or stool sample. These people work on poo, see? I don't have to do that. But they did find increased diversity. So the microbes that were there, even though they went in probably at 0.0001% of the total 100 trillion bacterial cells in the gut, had an impact on the other microbes around them and changed the balance of what was there. And this also came with enhancements in health in terms of decreased inflammation. So, I mentioned my daughter with ulcerative colitis. I definitely point her at this kind of thing and say, this is good data to base you eating more of my fermented foods that I make that you don't like. Anyway, so as the takeaway then from all of this, I would say, drink kombucha, enjoy its funky flavors. You're going to be ingesting some live microbes, and if you combine this with other fermented foods so that you're getting a diverse range of microbes that are live entering your gut, there can be a lot of potential health benefits there. But do take care if you are a two-pot screamer, because, as I said, some of the kombuchas can contain more alcohol than you expect. With that, I'll just say thanks again for having me here. This is a really cool idea and a cool organization. And I'd just like to thank my lab, and Sierra Nevada, and a couple of my alums on the left of the slide who were involved in kombucha research. Sierra Nevada, we did some work with, and they make a hard kombucha under the name Strange Beast that we helped them develop the culture for. So it tastes good. It makes no claims about health or probiotics, but tastes good. And my lab. So thanks again. I appreciate the invitation to be here and look forward to your questions.
Thank you so much. It's not often that we have scientists that are sponsored by Sierra Nevada. So I think that is quite possibly the coolest funding acknowledgement I have ever seen. So we're now going to enter the question and answer phase of tonight's Science on Tap. And I have a couple of requests. Number one is that your question ends with a question mark. Please and thank you. And number two is that we are asking single questions. It is in the grand academic tradition that we would ask 14 questions as one question. We want to make sure that we have a chance for everybody to ask a question. After I kick off by asking him a question, then I'm going to run into the audience, raise your hand, and then I will run around and I will hold the mic up to you so that you can ask the question, but I will still be holding the microphone. So please don't try and wrestle it out of my grip. Our insurance liability says I must hold this at all times. All right, so with that, I have a question for you about how the SCOBYs, as you transfer them from culture to culture, as you continue to brew amazing kombucha, how much does the microbiology balance internally change from culture to culture?
That's a good question, actually. It stabilizes, and we find that we did analysis on some SCOBYs from commercial producers 6 months apart, and they had the exact same microbes in the exact same proportions. It was pretty crazy. Every now and then you will find destabilization. That's usually because something else has changed. So if you do it consistently, then you're going to end up with a stable culture where the bacteria and yeast are cooperating, and they balance each other out nicely. Yeah. So surprisingly stable.
Excellent. Question here.
You were talking about how some of the kombucha was 4% that was claimed to be non-alcoholic. Did you notice from similar brands or brands but were sold in a different retail that there was a difference in that alcohol content?
As in, are there some brands that are kind of repeat offenders? Oh, okay, good question. Like some retailers who maybe aren't very good at keeping things cold. Yeah. Uh, I'll say no only because we didn't, I mean, we had a decent number of samples, but I wouldn't say enough to really make that kind of conclusion. Not from a, we didn't have any like, this all came from one retailer kind of scenarios where all of the like high alcohol ones were all from like one supermarket, but that could definitely happen for sure, yeah.
Question here.
The Stanford study, was kombucha considered one of the fermented foods?
Oh yeah, it was, yes. So in that Stanford study, the diet, they consumed quite a few different fermented foods and kombucha was one of them, yeah. And that was over, it was a 14-week study, and the differences that they observed in the gut microbiome and the inflammation markers were kind of starting to happen within about 6 weeks.
Question here.
So I've brewed beers, ciders, and even vinegar. And what I noticed in that the jar and showing what's going on is it looks like what's happening in kombucha is very much like what you're going to do when you brew vinegar. So what's going on different in tea or apple juice that's contributing to a different end result?
I mean, if you're comparing kombucha to vinegar, the main difference is usually you're going to carry the fermentation out in 2 phases. You're going to do the alcoholic part first, and then you still have a similar system where you've got a vinegar mother like floating on the top and it's making the acetic acid. The main difference is because all the sugar's gone, you're not getting some of the other organic acids. So when you taste kombucha and vinegar, they don't taste the same, and that's because it's not just acetic acid, which is what vinegar is. You've got acetic acid, gluconic acid. There's some others that are controversially meant to be there, but we never find them, like glucuronic acid, which is meant to be good for, I can't remember what health-related aspect, but we use very sensitive instruments to measure the organic acids, and we've never found it, so I'm not sure what's going on there. But that's the main difference. It's just that it's a parallel fermentation as opposed to, in vinegar, it's sequential.
Next question.
Are there specific microbes associated with the kombuchas that had high alcohol levels in the store and lower alcohol levels?
Yeah, that's a good question. No is the short answer. So most of the packaged, and the other problem, what I will say here is we're analysing after the fact. So what was causative in terms of the extra alcohol, we can't really say because it might have grown and then died off by the time we were sampling it. So I'm not sure we'd be able to make that conclusion anyway from the way we did this study. But no, there wasn't a specific type of yeast or bacteria that we found in the incorrectly assigned soft kombuchas.
Next question.
Hi. So with the controversy with its health issues, are there actually any clinical trials happening right now with kombucha? And if so, how are they sourcing it?
That's a good question. I'm not actually sure. I've certainly seen grants where people have tried to get money to do it, but I'm not sure if there are any large-scale clinical studies. The Stanford one was very good, but it was still a fairly small cohort of, I can't even remember how many. It was maybe 30 people or across the 2 groups. It wasn't a huge study, right? Yeah, I don't think so. I mean, there's more movement around the fermented food area in total, but I'm not sure, it probably wouldn't be too specific to kombucha. I think we'll probably see more studies like the Stanford one where, as a general idea of fermented food diets, we see this in NIH-funded studies where once there's a model diet for something, then there'll be a lot more research on it of like, all right, somebody's published, here's a way of asking the question about fermented foods, and it's not too specific to any one thing, and it's going to get at the bigger question. It wouldn't surprise me if we start to see more. I'm not aware of I haven't seen anything specific.
Opportunity.
Yeah.
Next question.
So you said that a lot of the companies just added bacillus to it. is there at least something special about kombucha that you can do that, or can you just add bacillus to anything you want and the industry jumped on it because health benefit?
Yeah, good question. Okay, so I mean, you could add it to anything. You can just, I mean, it's equivalent to going to Costco and buying an antibiotic supplement. I think two reasons why the bacillus was probably popular to add in this particular context, because it's not part of the SCOBY. It's relatively easy then to check that it's there, and it's a spore. Like a bacillus they're spore-forming bacteria. The spores are a little hardier and more likely to survive for longer in the bottle before carking it. So I think the survivability part is really important, and it's a well-known probiotic. So it could hit a few different reasons, but you could add it to anything. Now the question is whether, and this is an area where the research hasn't really fully developed yet, is the relative benefit of the same probiotic in a different context in terms of a food or beverage like kombucha. That probably have heard the terms prebiotic and postbiotic, right? As well as probiotic. So prebiotics are usually fibers and things like that. Postbiotics are a relatively new term, and that relates to metabolites that microbes make. This is kind of the kind of thing where I was talking about the gut-brain axis and the metabolites, communicating with your cells and doing crazy things. So in a matrix like kombucha, there can be potentially postbiotics that might accentuate the function. That might be different if you're just consuming the probiotic by itself as a capsule. But the research hasn't really been fully done on that.
Next question.
I realize it would be hard to kind of control for this because it's all going to be different, but have you or anyone in your lab looked at the difference between commercial kombucha and homemade kombucha?
Yeah, good question. I mean, yeah, my students make kombucha and like to, bring it in the lab and do some comparisons. So, in what sense? What kind of, like? Oh, okay. Yeah, not that part specifically. We've mostly looked at tasting them and deciding whether we find that, or what we started to find, and this has been something that's probably been happening over a few years. A lot of kombucha companies have kind of dialed back the funkiness a little bit. I don't know if you've noticed that, becoming a little bit more soft drinky and not so kombucha-y. And so we would then have homemade kombucha and go like, oh, yeah, that's right. This is what it used to taste like. And it's crazy how fast that's happened, but I've seen that happen with a few brands where they've really tried to hit a more mainstream flavor palette. But from a benefit point of view, not too sure.
Next question.
So if you had moved back home to Australia to open up your own company, what's the major limitation between moving from home brewing to scaling it up to industrial scale?
Yeah. Well, this comes back to the issue of if you're home brewing and you make a kombucha that's a little bit alcoholic, you're only really affecting yourself, right? If you're making it commercially, then immediately you're having to worry about, am I making something that's going to get me sued? And so you're going to have to be a lot more stringent on your controls. So you're immediately having to analyze things that, in a homebrew setting, you're just tasting, right? You're relying upon, okay, this tastes as acidic as I remember it last time. I think it's good. A lot of kombucha brewers who start out commercially still rely upon taste and feel, and that's when they can get into trouble. So first things first, you have to start analyzing stuff. Then as you start to scale up the size of the container in which you're fermenting in, you have two new problems. One is just you suddenly have a massive amount of SCOBY, right? Where you can't possibly keep it all. You can't put it all into a SCOBY hotel. And if you have a really big tank, they're quite large. It was Chelsea who was telling me the story before about the pile of, possible pile near Brew Doctor at some point of used SCOBYs. And their tanks are, probably a quarter of the stage at least. So the SCOBY's about a quarter of the stage and probably like this thick, and a stack of them, and people getting a little bit freaked out by this massive cellulose. So that's a new problem that you don't have as a home brewer, is suddenly you've got a lot of SCOBY to deal with. They're probably the two main ones, right? And then, the capital involved, okay. And really to control ethanol, at the end of the day, you really ideally need to have enough capital investment to be able to have a way of getting rid of the ethanol when you inevitably have a batch that you end up with too much alcohol. And that costs a lot of money. But they're probably the main challenges.
So
Plus I'm too, risk averse to actually do that, so I'll probably keep being an academic.
So as our last question, I would like to know, how popular is your lab on the day that you bring all of the kombucha samples in?
Very popular, particularly on the hard kombucha days. Then, the grad students definitely, pay attention to what's going on and when there might be some free stuff around. We also did a project on cheese at one point, and we ended up with a box like this big full of packages of Tillamook cheese that they sent us 2 boxes, and one we used entirely for the experiment we were doing, and the other box, and it was all different varieties, all of their different products. And we didn't need to use, and we said, do you want us to send them back? They said, oh no, the grad students can have them. So we're very popular that day too. Yeah.
Well, I think that your lab sounds like it's the absolute place to be. Cheese, kombucha, that's living the best life. So I want to thank you so much for sharing your expertise about this amazing field of kombucha with us today. So let's give Dr. Chris Curtin another round of applause. Thank you so much for attending tonight's Science on Tap. And as a reminder, our next one is going to be September 13th at the Kiggins Theater in Vancouver about eco-crossings. We're taking a break for the month of August, but again, we would love to hear from you on that feedback survey. Let us know what kind of topics you'd like to learn more about, whether or not we should invest in a kombucha company with Dr. Curtin here, and any other thoughts that you may have. And on behalf of all of us on Science on Tap, thank you so much and have a lovely night. For those of you who scored a 6 and above, come up to the front of the stage and grab your awesome pint glass and have a great evening.